Hello viewers, in this particular lecture on bacterial sexual reproduction, I shall be touching upon the different aspects of sexual reproduction in different prokaryotes other than bacteria, like cyanobacteria, Rickettsia, Chlamydia, Mycoplasma, Actinomycetes. And then I shall also touch upon the different mechanism by which a particular cell is getting competent to receive the foreign DNA so that it can be transformed. And naturally, the first group which comes is the cyanobacteria. Now, the cyanobacteria commonly is known to have no sexual reproduction. There is only asexual spores developing. But in a particular organism, as you can see, this is Amycolaopsis. In a particular strain, there are different types of projection in the cell. These projections are actually similar to pili or pseudopili. And naturally, this particular organism is also important because they are photosynthetic, meaning they are capable of photosynthesis, and at the same time, they are also surviving as saprophytic organism. So there must be a unique dual combination playing in this particular organism. So it was suggested that this particular stable photosynthetic protein and heterotrophic growth must have been accounted for by the presence of some pili or pseudopili like structure which was actually responsible for their transformation. Now coming to this particular structure, definitely it was indicated that they have the pili like bacteria. And subsequently, it was observed that the pili protein like the B, C, D, G, H, M, N, O, P and even COM-A, they are all present in a sequence-specific manner in this Amycolaopsis strain number PCC6803. So naturally, this particular Amycolaopsis is capable of showing transformation. And at the same time, there are also specific putative genes which are present in this particular organism. It was observed that when a particular type 1 endonuclease was damaged, they were showing transformation at a faster rate. And this faster rate meaning almost 5 times greater than the normal rate of transformation. So normally this particular pili or pseudopili, which is otherwise very rare in cyanobacteria, is present in Amycolaopsis. And you can see in this particular slide, there is typical retracting type 4 pili in cyanobacteria like other gram-negative bacteria. And this type 4 pili, they are growing like in pilin monomer, they are growing one after another and they are producing this long pili. And at the same time, they are helping in the movement of the cell against a solid substratum. So when they are polymerizing, they are attached to the substratum, and naturally when they are depolymerizing, the cell is moving. But this is against a solid substratum. If it is against two cells, then naturally the cell might come close to each other. Not only that, there is also another structure which is present in the form of a pseudopilus in between the outer and the inner membrane. And this pseudopilus is also energy driven and is also helping in the reproduction or in the transformation phenomenon in this particular bacteria.
Sir, I have a question.
Yeah.
What is the difference between a pseudopili and a pili?
As you can see that this particular pseudopili and pili both are present on the bacterial membrane. But the pili, as you can see, is projected outward. The pili is projected outward. So naturally, it is helping in the direct transformation or direct attachment with another bacteria or a cyanobacteria and is helping in the exchange of gene. But in case of pseudopili, definitely it is embedded, and naturally this pseudopili is maintaining coordination and between the outer and the inner membrane. This coordination helps in the purpose of transformation, that is, it is acting as an associated structure for transformation. Clear? Now coming to another interesting cyanobacteria, which is Anacystis nidulans. Now this particular cyanobacteria is thermophilous in nature. There is a plasmid called PCH1. This plasmid can actually be incorporated in this Anacystis and it will show resistance to ampicillin. And this particular transformation can take place within 15 to 18 hours. So it is pretty quick. And at the same time, it is at a very low dose, this transformation is possible. Similarly, there is another particular plasmid which is PSG111, which is also accepted by an anacystis, and it exhibits the resistance to chloramphenicol antibiotic. So thereby we can say that apparently there is no sexual reproduction in cyanobacteria, but because of this particular transformation plasmid, they are definitely changing their morphological or changing their physiological pattern. And there are some factors which definitely increases the transformation rate in the cyanobacteria. It can be pre-exposure or pre-incubation in darkness. It increases the rate of transformation. It can be sometimes photosynthetic process. If it is blocked by some blocker like DCMU, then the process of transformation increases. That means there are some physiological change, and at the same time, if the cells are grown in iron deficient condition, then the cell will show transformation. So naturally we can say that cyanobacteria under some environmental stress will show faster rate of transformation. But what is the application of this transformation? The application include, number one, definitely it will be internalizing some foreign gene. So it will acquire some property which is not there in other organisms. So that is, it will gradually, it will show evolution, it will evolve to a better way. And at the same time, because of this pili, it can colonize. And at the same time, it will actually modify itself. Say for example, through the incorporation of photosynthetic gene, it becomes photosynthetically more productive. Chlamydia or Chlamydophila, as it is commonly called, is not actually a pure prokaryote. It was considered initially to be similar to virus, but later on it was proved that it is in between virus and prokaryote. So it is a connecting link between virus and higher organism. So this particular Chlamydophila or Chlamydia, they can invade different mammalian host, birds, human even, and they exist in two forms. You can see they are remaining as elementary body, and they enter into the cell and gets vesiculated, and they produce what is called reticulate body, and then again they produce the elementary body within the host cell, and these elementary bodies are again released outside. So it is a unique alternation between elementary and reticulate body, but there are no other cellular organelle. Now this particular organism of Chlamydophila, they may have different species, like Chlamydophila pneumonia, which is invading human. They are having a particular CPAR39 gene fragment which was believed to originate from a phage virus. Similarly, there may be Chlamydophila cavi, which is invading other mammals, is having sigma CpG1. At the same time, there may be Chlamydophila parcorum, which is attacking mammals again, is having CHP3. What are these? These are gene fragments. Similarly, there may be Chlamydia psittaci, which is invading the birds, is having CHP1. And at the end, there may be Chlamydophila abortus, which is also invading mammals, is having CHP4 genome. And this particular CHP4 phage, you can see in this particular slide, is donating a 4530 base pair gene fragment. And the advantage of particular gene fragment is that it is having multiple ORF, that is open reading frames, and at the same time there may be variable insertion sites. So what does it mean? It means that as we have suspected that this particular Chlamydophila is not purely prokaryote, it has originated from virus. So this particular observation of the presence of virogene in Chlamydophila indicate that they have taken up certain genes from viruses because of their close similarity, which initially might have started in the form of a infected organism, infectious organism, but later on it was actually taken up by the cell or internalized by the cell as a cellular genome. So this type of transformation, what is the immediate application? Naturally, I can say that it imparts pathogenicity, invading host. At the same time, it is definitely showing a pathway of evolution from virus. And at the same time, it is also by the presence of these particular plasmids, we can give an internal classification to the Chlamydophila organisms. Mycoplasma is a group of organism which is considered to be the smallest prokaryote which is having a proper cell. but it is not having a definite outer wall, so it can change its shape. But this mycoplasma is also capable of showing faster transformation, and this DNA normally is accepted through the membrane outside. This particular Mycoplasma pulmonis, which is a very common cause of pulmonary infection in mammals, is capable of showing transformation through some plasmids. These plasmids may be PAM120, these plasmids may be TN916, and naturally, because they accept the foreign particular DNA, they can modify themselves. And naturally, this group of genes may be existing in different Mycoplasma species. Like there may be gene fragments of Mycoplasma pulmonis present in Mycoplasma hyaerinis. So naturally, this intraspecific transformation is very common in Mycoplasma. At the same time, when we actually see that what is the nature of this plasmid, say for example, you can see this is TN916, which is a plasmid having approximately 18,000 plus base pair. And they are having specific nick sites. But what is important, there are some areas which is, say here it is in between 20 and 21. This area is responsible for taking in other genes, and this area is modifying the plasmid, and it will make a non-conjugating plasmid to a conjugating one. And most important in this TN916 is the left side. In the left side you can see there are specific broader areas like the TRA and also the different antibiotic resistant genes. So naturally the TET-M, which is conferring resistance to tetracycline and minocycline, they will definitely show that they will act as a marker gene, and successfully the gene has been transferred. So thereby we can say by this transformation mechanism, the Mycoplasma can become resistant to a particular antibiotic, and naturally this will impart their pathogenic nature. So what are the applications of this particular sexual reproduction or transformation? Definitely we can say in the manufacturing of integrating vectors or shuttle vectors, which will shuttle between two organisms. And at the same time, it is definitely showing a particular evolutionary pattern, that is whether evolutionary it is advanced or primitive. And at the same time, it is giving pathogenicity, because they are all infectious organisms. And definitely this evolution will be towards antibiotic resistance, which is not a good news for us. Rickettsia is an organism which is causing the common typhus fever. Now Rickettsia prauseki, which is the common causal organism for typhus, shows resistance to rifampicin antibiotic. And this resistance is due to a particular gene which is the RPOB, and this RPOB gene is getting mutated. It is getting mutated by arginine lysine replacement or point mutation in 546 polypeptide position. This particular RNA beta subunit or RNA polymerase beta subunit is getting modified, as a result of which the particular organism becomes resistant to rifampicin. Now naturally this rifampicin resistant gene can be transferred through a particular process of transformation, and naturally it will increase the infectivity of Rickettsia. And you can see that Rickettsia infection can be very commonly denoted by some technique called fluorescent in situ hybridization. You can see in this slide that the fluorescent green color is coming from the Rickettsia cell, and this Rickettsia is invading the Glossina morsitans organism, male Glossina, which is a common arthropod. So a particular Rickettsia infection is definitely a common phenomenon in arthropod, and you can see that how much colonization that Rickettsia is carried out. And at the same time, when you can see in this slide, the uninfected cells are not showing these green colonies of Rickettsia. So FISH technique is a very common technique to detect Rickettsia in a particular host cell or host cell culture. And Rickettsia is actually a successful parasite. It is definitely not causing infection only within a single host. It can go into another host, and there are certain vectors which actually carry them. And this particular rickettsial genes is also present in E. coli, so which actually suggests that Rickettsia is not limiting itself within its associated species like Rickettsia or Coxiella, etc. It is also going into other bacterial organisms. Now there are definite barriers towards this gene transfer. Now what are these barriers? First of all, it is definitely an intracytoplasmic pathogen. So that way it is limited, it is not growing, it is not going to different other organisms to a great extent. Its mutation rate is low. So if I am stable extracellularly, what is the need of getting into another organism? I will be stable outside as a non-pathogen. And at the same time, there are plasmids present but less in number. But even then, considering all these bacteria, we also have seen that how Rickettsia is percolating as an infectious organism. Apart from the particular phenomenon of fish, Rickettsia can also be detected by a unique transformation where they have used this particular RPOB gene and they have fused it with a green fluorescent marker or green fluorescent UV marker plasmid. This particular green fluorescent marker is being fused with the RPOB. So apart from this rifampicin resistance, we can easily detect that whether the cell is getting induced or whether the cell has entered successfully by the unique facts or the fluorescent activated cell sorting. You can see in this particular beautiful slide, all these cells are Rickettsia cells where there are expression of the GFPUB and the RPOB gene. So what are the different applications of this transformation? I told you it will definitely indicate a specific infectious stage or infectious nature of Rickettsia, and at the same time it will also help Rickettsia to survive better within and outside the host's body. Actinomycetes is a group of organism which is in between prokaryote and fungi. Actinomycetes, you can see, this is a filamentous structure, and this particular organism is called Amycolatopsis. So Amycolatopsis is a particular organism which actually is UV sensitive, and at the same time it produces an antibiotic like compound which is epoxyquinomycin C, which is definitely a broad spectrum antibiotic. This organism is producing a plasmid which is the PMEA 300. This PMEA 300 is approximately a 29 kb plasmid and is definitely transferred from one host to other. And as it goes, it will definitely carry these properties of resistance to UV or UV sensitivity in the better way, and at the same time it will also produce the tyrosine or other amino acids, and at the same time it will produce the antibiotic. So transformation in actinomycetes is also used to study evolution, production of secondary metabolite, and at the same time UV sensitivity. And of course, when I say tyrosine production, tyrosine is definitely used in the later production of some hormone. You know, I am talking about the thyroid hormone. So naturally this transformation of genes will be useful in the human development at a later stage. The mechanism of DNA uptake in this particular bacterial cells or bacteria related cells. Most important phenomenon is whether the cell is ready to accept the DNA, and this phenomenon is called competence. So regulation of competence is very important. Now, definitely I have said that this mechanism of acceptance of DNA depends on certain protein present in the membrane. And it can be that in gram-negative bacteria, it can be type 4 pili, which is having a typical retracting property or adhesing property, which will bring in two cells close to each other and thereby will accept the DNA. And there is also another protein like the PIL-Q protein, which will help in the transformation. Similarly, in gram-positive bacteria, there will be the pili and the pseudopili, like in cyanobacteria, which is helping them to transform. But what are the common features? There are certain proteins which are unique, like the COM proteins, the COM-EA, COM-EC in Bacillus subtilis, or COM-E in Neisseria gonorrhoeae. These are common. And this particular transfer is ATP mediated in both, because I have shown that there are specific ATPase at the base of the pili. And at the same time, there will be the typical recombination which will be brought about by the Rickettsia system. So coming to this unique system of transfer in Neisseria gonorrhoeae, you can see that this is the DNA which is accepted at the PIL-Q level, and then finally it goes into the pili, and then through COM-P comes to COM-E, and then to COM-A, and finally into the cell. So this is how this DNA is transferred. But when we see this unique Streptococcus pneumoniae, which is a gram-positive bacteria, how this competence is being regulated, or how this competence is brought about, because the cell must be prepared to accept the DNA. So what happens here in case of this Streptococcus pneumoniae, there is a typical competence stimulation or stimulating peptide, CSP, and this from this pre-CSP or from this COM-C, it will be going out via the COM-AB channel outside. The CSP is going outside and will be accepted by some, and this, this ABC transporter is having the CSP outside and it is accepted by COM-D. And now what is this COM-D? COM-D is actually a particular receptor which is histidine kinase. And the moment it is accepted, it is now getting phosphorylated. And this is being transferred to COM-E. And then finally, this COM-E phosphorylated will be giving it to COM-AB, and then to COM-CD, and then finally to COM-X. So this is a unique way by which as many as 60 genes are activated. And ultimately, they will lead to the production of alternative sigma factors, as a result of which the DNA uptake will be possible. So you can see the unique signal transduction, which is starting with CSP or pre-CSP and finally ending with the transcription of those competence factors which will allow the DNA to percolate inside the Streptococcus cell. In Bacillus subtilis, the transformation is initiated by a different mechanism of competence. Here we can see in this particular slide that there is the PHRC, which is transporting or exporting the pro-CSF, that is competence stimulating factor, and then it is finally recognized by the simple permease, which is getting inside the cell. And at the same time, there is another one which is the COM-X. COM-X is also exported outside, which is a pheromone, and then again it is accepted by a typical COM-P or the histidine protein kinase. And both will interact and will ultimately activate the COM-AP. At this point, there may be the RAP-C, which is inhibiting the binding. So naturally, this will reduce the rate of competence development. But once the COM-AP is activated, the transcriptional factor will be active, and in a sequential manner, the different DEGQ or the RAP-C or the RAP-A, which is blocking the process, which will be overcome, and finally the COM-S will be active, and there will be the genomic competence. So Bacillus subtilis, there are two specific pathways through which this particular competence is active. But at the same time, there are some specific common areas where we can say that both have the COM protein which is responsible for competence, which may be COM-AP or COM-EP. At the same time, the component may be exported outside and then recognized from outside because of the development of the gradient in the outside the membrane. And finally, this transporter protein is membrane oriented, which can be ABC transporter or it can be PHR protein. So these are the common areas between Bacillus subtilis and also the Streptococcus pneumoniae, though both are different bacteria. In case of Haemophilus influenzae, we have a different type of competence. We see when this particular organism is grown in starvation media, then this particular organism will develop the competency. And this is mediated by the CRP-SXY regulation. And the CRP is same as the catabolite activator protein, which is regulating transcription. There are definite promoter elements which will activate the CRP and which is definitely dependent on the CAMP concentration. And what happens is whenever ultimately the CRP is activated by SXY, then there will be a unique curvature of the RNA polymerase, which will ultimately bring about the transformation mechanism in Haemophilus influenzae. And this particular SXY CRP system is also known in other organisms, like in Vibrio cholerae, where we have the unique TFO genes which are similar to SXY, helping in competence. Now we come to the end of this particular lecture. In this particular lecture, we have discussed the different ways of sexual reproduction in other forms of bacteria or other forms of prokaryote, like the Chlamydia, the Rickettsia, the Actinomycetes, or the Mycoplasma. And we have also discussed the mechanism by which a particular bacterial cell will acquire competence and how this unique signal is actually taking place, or signal transduction is taking place in the bacterial cell, so that the bacterial cell become competent or ready to accept the DNA fragment from outside. Okay?